I'm here with Dr. Valerie Vanhoorne, who presented on the important topic of continuous manufacturing during a two-day symposium in Ludwigshafen. Dr. Vanhoorne graduated from Ghent University in 2011 as a pharmacist, and since 2016 has been active as a postdoc in the Laboratory of Pharmaceutical Technology, with a focus on continuous direct compression and twin screw granulation. Welcome.
Thank you. So continuous manufacturing is defined as a method to manufacture, produce or process materials without interruption, hence making the most amount of product in the least amount of space and time. And this is in contrast to batch manufacturing, where we will typically process a specific amount of material. So we will load a specific amount of material at the start of our process. Then we will process it for a specific amount of time, and after we reach the desired end quality, we will discharge it from the process.
Could you briefly go through your presentation, highlighting the key points, please?
Okay, so my presentation, I introduced the principle of continuous manufacturing, especially highlighting advantages of continuous manufacturing over batch manufacturing, which is still prevailing in the pharmaceutical industry. Then I also explained how process analytical technology goes hand in hand with continuous manufacturing. And then I gave an overview of where we are today, which drug products are on the market, which are continuously produced, and also about manufacturing technologies that are available nowadays. And then I proceeded with a case study on twin screw granulation. In the first part of the study, we focused on twin screw granulator itself. And then in the second part of the study, we used the entire continuous line using the granulator, dryer, mill, and tablet press. So therefore, the aim of our study was to investigate if this polymorphic transition would also take place during twin screw granulation, especially since during twin screw granulation, the residence time inside the process is much shorter. So during twin screw granulation, the residence time distribution is in the order of 5 to 20 seconds, whereas during high shear granulation, the residence time inside the process is in the order of tens of minutes. So then some intermediate conclusions on this part of the study. So first of all, we could see that the granulation behavior of delta mannitol was different in comparison to granulation, twin screw granulation of other excipients such as lactose and microcrystalline cellulose. The polymorphic transition from delta to beta mannitol also took place despite the very short residence time and despite the typically lower liquid to solid ratios we apply. However, the polymorphic transition was dependent on the process parameters, and the most influencing process parameters were the screw speed, liquid to solid ratio, and the number of kneading elements. And we obtained excellent tabletability after the polymorphic transition, which could be attributed to the higher specific surface area and also to an increased plastic deformation. So therefore, in the second part of the study, we performed trials with a formulation consisting of 75% of paracetamol, which is known for very poor compaction properties, and 25% of delta mannitol. And we varied the most influential parameters we identified before on the polymorphic transition. So the conclusions of this study were that the transition also took place, even in combination with the highly dosed API. The transition was mainly influenced by the liquid to solid ratio during granulation, but not by subsequent drying or milling. And that granulation with delta mannitol could overcome the poor tabletability of paracetamol, while at the same time producing also easy flowing granules, which is the aim of our granulation process.